Hi guys, this is the lecture for mitosis, and mitosis is basically just general cell division. We take one cell and we literally just split it into two. Some would call it just making a clone. So mitosis really results in two identical daughter cells. We will replicate the DNA exactly, so each cell will actually get the same DNA and everything. So it involves your somatic cells, which are all your body cells, with the exception of your sex cells, what we call gametes, so your egg, ova, oocytes, and then obviously your sperm. And it's used to replace damaged or dead cells and dying cells, and then also to add mass to a growing organism. So taking it from conception, having those that fertilized egg replicate and replicate and replicate and divide and divide and divide, and then obviously going from a baby to an adult. One note with that would be I'd like to talk to you a little bit about cancer. Cancer is basically uncontrolled mitosis or rampant cell division without a purpose. So it doesn't need to divide because it's replacing damaged or dead cells. It doesn't need to divide because it's trying to increase the mass of an organism and so on. So there's no purpose for that cell division and it just runs rampant. That's cancer. But I want to go through just general cell division with you. Normally during a cell's life, a cell spends most of its life in what we call interphase. And interphase is not part of mitosis. Interphase is where the cell grows and it metabolizes, so it catabolizes, it breaks down molecules, and then anabolizes, so it actually builds up other molecules, so it just lives. And then at one point in time, basically a purpose for its division comes up. So maybe it does need to add more mass to an organism, or maybe it does need to replace dead or dying or damaged cells. Regardless of that, that purpose does come up, and at the end of interphase, the DNA, so all 46 chromosomes, and I'll just put a little X for chromosomes, but the 46 chromosomes are going to be replicated. So they're going to be copied, and they're going to actually double. So now that one cell is going to have twice as much DNA or genetic material in it. And the reason being is that, like I said before, we're going to take this cell and we're going to literally just split everything. So the other thing that also needs to happen while DNA is being replicated is that any, especially vital organelles within the cell, also need to be replicated, especially if there's very few of them. So the cell will also do that unless there's enough of them. But that's at the end of interphase. And then the cell is really ready to go into mitosis. So let me just bring this up real quick. Mitosis, as you see on this little chart here, this should look very familiar to you. This is only one part of your meiosis huge long page that I gave you a couple of weeks ago. But mitosis really involves prophase, metaphase, anaphase, and telophase. Those are the four major phases of mitosis. And you see that I have this nice little dotted line down here dividing these four mitotic phases from cytokinesis. The reason being is that cytokinesis isn't really officially part of mitosis because, by definition, mitosis is the division of the cytoplasmic contents and DNA, or genetic material, whereas cytokinesis is division of the cytoplasm as well as the plasma membrane. The other reason is that some cells actually don't get to cytokinesis. They'll go through prophase all the way down to telophase, but they'll stop at telophase. Your liver cells are an example of that. Liver cells, you'll often see multiple nuclei. If there's two nuclei in one cell, so if we've got a cell and we've got two nuclei with a bunch of genetic material in it, we say that that is binucleate. Okay, now let's say that maybe this cell has gone through mitosis a few different times and it's got all these different nuclei in it. Okay, then we call it multinucleate. Okay, so if it's bi, it's got two. If it's multi, it means it's got more than two nuclei in it. And sometimes you'll see that, especially in your liver cells. So that's one big reason why cytokinesis really isn't part of mitosis. So let's just go through each of these phases I'm going to make this a little bit bigger. All right. And I've got some notes for you on the side here, so maybe I can make this a little bit bigger. All right, so during prophase, what you're going to be seeing is the NE is the nuclear envelope. So the nuclear envelope is going to begin to break down. So here's our nuclear envelope, and it's going to begin to break down. And the reason that it really actually needs to begin breaking down is because chromosomes are trapped inside of them. And during this portion of prophase, you're going to see chromosomes begin to condense. And what that means is right now they're in the form of what we call chromatin. And chromatin is basically, kind of looks like spaghetti and meatballs. It's basically unwound yarn of DNA in no particular shape or form. And then you also get to see what are called histones, which are just a grouping of specialized proteins. So it kind of looks like spaghetti and meatballs. So during this time, chromatin actually will condense into those nice little X shape, that we normally associate with chromosomes. And they're stuck in the nucleus. So it's really imperative that that nuclear envelope breaks down to kind of release them. The other thing that's going to happen is that centrosomes, and hopefully you remember those from when we studied the cell, centrosomes are actually going to migrate so that they are directly opposite from each other on either side of the cell, okay? And if you remember anything about centrosomes, centrosomes help produce microtubules, and that's going to play a huge role in basically building up that mitotic spindle, which helps to direct mitosis. And remember, if you remember anything about microtubules, those are structurally sound. They're highly used in various areas of the cell to give it structure and shape because they're very flexible, but they also help keep the shape of the cell, especially under pressure, and it allows it so that the cell doesn't actually get torn apart. So the centrosomes migrate to opposite ends of the cell. Chromosomes form and the nuclear envelope breaks down, and at that point then the cell should be ready to go into metaphase. Now, one real quick thing that I do want to go over with you is basically the form of a chromosome, and I'm hoping that you remember this from advanced biology, but a chromosome is made up of two strands of DNA. There's one and there's two. And these two strands are what we call sister chromatids. When they're put together, they come together and they give you that nice X shape, okay? And there's something called a centromere. Write that over here. A centromere, and that's this little button guy right in the middle. That centromere is responsible for holding those two strands of DNA or sister chromatids together. Now, it's kind of confusing because sometimes we consider just this one strand, so by themselves, we will call them chromosomes. We'll call this whole thing a chromosome. So, you know, I guess just kind of pay attention to what you're looking at to kind of determine what we're talking about. But that's really the structure of a chromosome, and that's going to play a big role in a second here. So at this point, we have gone on to metaphase. And metaphase is kind of fun because metaphase deals with this line. This is an invisible line in the cell, and it really determines the spot where the cell will divide right in half. And we call that line the metaphase plate. Okay, so it's metaphase and then plate. And it's not a visible line, it's invisible. It's just an area where we can see under the microscopes those chromosomes will actually line up directly on that metaphase plate. And usually the centrosomes are always on the opposite sides. And there are the mitotic spindle fibers. And the shape those fibers make, and there'll be more over here and so on, you know. But that overall shape, that's your spindle forming. And what those fibers are actually attaching to are the centromeres of the chromosomes. So chromosomes line up on the metaphase plate, and the spindle fibers attach to the centromeres. And that's all that really happens during metaphase. So let's just move this up a little bit. Okay, now we're at anaphase. Anaphase basically is kind of the fun one because then you see, there's your centromeres, then you're going to see those chromosomes literally be torn apart. Okay, so here is 1/2 of a chromosome. So that's one piece of DNA or one sister chromatid. Those are your spindle fibers. And basically those that spindle fiber, or the spindle itself, is going to direct those sister chromatids to opposite ends of the cell. So they're actually going to get ripped apart. So the sister chromatids are going to be separated from each other from their centromere. So they're going to get ripped apart, and then they're going to be driven to opposite ends of the cell. So fibers pull chromosomes apart, and they're pulled to opposite ends of the cell. Now, the reason being is that this middle part is like the danger zone, okay? That's still kind of where the metaphase plate was, okay? It's that area that marks where cell division is literally going to occur. So we don't want any organelles and definitely no genetic material caught in this area when cell division actually is going to start splitting the cell in half, okay? So let's just move this up a little bit. And let's see if we can't move. All right. So now we've gotten to telophase, and a couple of different things happen. So now you've got all of those chromosomes. Again, I'm referring to them as chromosomes floating around. Okay, and one important thing is you have 46 on this side and you have 46 chromosomes on that side, because remember, we replicated them and doubled the amount that we had so we could split them exactly in half. Okay, so those chromosomes are literally just hanging out on opposite ends of this cell. So what's going to happen is that there's that Ne again, okay, so that nuclear envelope is going to start to reform around those chromosomes, okay? So you're going to see that this one cell, okay, is actually going to become binucleate for, not even a second, but for a short period of time, because it's got those two nuclei in them. So the nuclear envelope is going to reform around those chromosomes, and what you're going to see is those chromosomes are actually going to decondense back into spaghetti and meatballs, okay, or back into chromatin, okay. The other thing that you're going to see too is that cleavage furrow, and this is right at the very end of telophase. It actually marks the end of telophase and the beginning of cytokinesis, but this cleavage furrow right here is going to start to form, and it actually forms exactly where that metaphase plate was, because again, remember, that's where it marks the literal cell division or separation of that cell. The other thing you're going to notice, okay, is that your mitotic spindle is going to break down, and those microtubules that acted as spindle fibers are going to be reabsorbed into the cell somewhere and used as, part of the structure of the cell somewhere. So that's telophase. And again, sometimes your liver cells only get to telophase and they don't go on, but that's mitosis, okay? Now, if the cell actually is going to divide in half, okay, what you're going to find is literally that cell is going to pinch apart from itself, okay? So it's literally going to pull away, and this right here is going to get really, really small, okay, to the point where it just breaks apart and it pinches off, okay? And what you end up getting are these two completely identical daughter cells, okay? And each of them have 46 chromosomes in each cell. So we literally copied it and made a clone. And that is mitosis in a nutshell.